Blog Talk Radio.
Today's guest is Dr. Liu. His first name is Dr. Dongyu Liu, formerly a professor at the College of Veterinary Medicine, Mississippi State University, who has published a number of textbooks on PCR screening of food samples for human pathogens. Dr. Liu is now a resident of the country of Australia and is now engaged in other research. Dr. Liu, can you update us on your current projects?
Thank you, Andy. I spent 6 years at Mississippi State University College of Veterinary Medicine. While there, I did research on molecular detection of foodborne pathogens, especially Listeria. Listeria is a very common foodborne pathogen which occurs commonly in ready-to-eat food and all the fresh products, unless it's detected early, and it can be potentially very dangerous. And there have been instances where people are eating just uncooked or not fully cooked meat and poultry products and get sick, and even some of them even die of the diseases. In 2008, I moved back to Australia, where I'm working on molecular detection of emerging infectious diseases, such as biosecurity organisms. And I'm the editor of a book called Molecular Detection of Foodborne Diseases, Pathogens. Okay.
Very good. Dr. Liu, can you identify for us the various types of traditional methodologies food scientists traditionally use to detect E. coli, Salmonella, Listeria?
Yes. Traditionally, laboratory culture is used for detection of foodborne pathogens, and the difficulty of the culture, the organism, and the low sensitivity of this traditional method means the results will probably know in weeks. Early, probably in 2 weeks. That's you really need to get the definitive results. So there have been a recent development in the molecular detection method which allow you identify a foodborne pathogen in 1 day or two, and which is much better improvement of the traditional method.
So,
okay.
As we look at the PCR method, what is the specificity sensitivity of PCR polymerase chain reaction as compared to traditional culture methodologies?
In terms of the sensitivity, PCR has the ability to detect one organism, and the specificity is 100% because the primers, the genes you're selecting, are only present in the organism you're detecting. On the other hand, with the traditional method, like culture-based methods, you will be lucky to detect 100 or 1,000 organisms. So, in some food samples, you may have under 100 organisms, and you probably wouldn't be able to detect. Okay.
Dr. Liu, I hear from a number of my food clients that they're challenged by PCR because of the live versus dead analysis question. Can you address that for a minute?
Yes, I can see there is kind of perception that that PCR is only able to detect the dead organism, but actually there is a modified PCR method which detects the RNA of the foodborne pathogen. So because RNA is only produced by live bacteria, so if you are using a test targeting the RNA of the pathogen, you should be able to detect the live organism. Another method that you can use to detect the detect or separate dead and viable organisms that using a kind of special kind of primers or stain, which will only recognize the viable organism, and we will not give a result with the dead organism. So there is a way to improve, or if you really want to, there is a way to detect the viable organism by PCR.
Very good. There's also a discussion about the presence of inhibitors in samples that, for example, with fruit and vegetables, the color of the sample may inhibit the PCR reaction.
Yes, that's right. Actually, there are more than a number of kind of inhibitors in food specimens, especially like the meat specimen, or like milk or cheese. So there's a challenge to use a kind of sample preparation method which will allow you to get rid of most of the inhibitors without sort of removing the organism in the samples. Okay.
Very good. The issue for most food producers is the dilemma of producing their fresh products, which do not have a kill step, and essentially shipping them out while waiting for the traditional culture results, or to run a PCR test and know with a general certainty that the product is relatively safe for shipment. Would you like to comment on that protocol?
I believe this protocol is generally working well in most of the cases, and if you have a large number of bacteria on the food sample or on the food products you're going to send out, and it will be detected and it will be kind of eliminated, or you are not going to send out this one. On the other hand, there may be some, most of the cases, they are just small number of bacteria, you are unable to detect, and because you are using like a very precaution in storage and all that, it wouldn't be a problem. The only problem is that when consumers buy the products and they are not following the guideline, like put the food in the 4 degrees, or some people may have a fridge which are out of sort of like it's not precise, and they think their fridge is like 4 degrees, but in fact it's probably 8 degrees. In this kind of cases, you may have some samples, or some food stuff you leave on the fridge, and some small number of bacteria may be able to survive. And Listeria is one of the examples which has the ability to survive at 4 degrees and grow slowly on anything up to like 10 degrees, not a problem. So that will be the case that will be a potential source for problem for the consumers.
Yes,
okay.
I agree with you. And your expertise in Listeria is particularly apropos because many of my clients who are food producers go to great extremes and expense to keep their food production areas cold, 38 to 42 degrees, and yet it's a prime area for Listeria. Can you comment on that?
Yeah, Listeria is kind of peculiar bacteria because on the surface, or just when you look at it, it doesn't look any different from other bacteria. However, this bacteria has the ability to grow and really survive at low temperature. Most other bacteria probably wouldn't be able to grow at 4 degree or 8 degree temperature, but Listeria can maintain and grow in that. And although it's a slower growth, but it's definitely growing. So that's why Listeria tends to be one of the most detected organism, or most relevant organism which cause foodborne diseases in humans because of its ability to survive at a low temperature. Okay.
So with those parameters of survival at low temperature, it would seem appropriate for food producers that are producing lettuce or fresh green leafies to perhaps capture the water sample of their wash bath and then run that as a sample of PCR for the presence of the organism.
Yes, it's definitely useful to screen the waters you're going to use to prepare your say lettuce, and also there'll be you'll probably be useful to screen the surface samples like the bench top and all that. So in doing that, you reduce the chance of this bacteria getting into your food product and causing problem later.
And your expertise in the area of multiplexing would lend a great deal of credence to the idea of developing a multiplex assay for E. coli species, Salmonella species, and perhaps Listeria species. Wouldn't you agree?
Definitely. I mean, the advantage of multiplex PCR is that you're able to put multiple primers which target the distinct genes from different bacteria. And by running this multiplex PCR, you can get multiple results just in a single run. Instead of if you run in separately, you take 3 or 4 tests to get the results. Okay.
What are the challenges with the multiplex assay, particularly with inhibitors?
I think the challenge with multiplex assay is really the challenge of a single-plex assay. I mean, if your sample contains inhibitors for one organism, it will also inhibit the other organism or the other assay. So essentially, if you are able to develop a single-plex PCR, say for example for Listeria, and you show that the sample preparation method you're using is helping you to get rid of all the inhibitors, then I don't see this sample preparation method will create the additional or separate challenge for the other tests within the multiplex format. Okay.
So what you're saying, Dr. Liu, is that with an appropriate extraction protocol, the sample can be prepared to minimize the effect of any inhibitor reaction.
Definitely. I mean, actually, this sample preparation method, I feel, is probably more critical and it requires more attention to than just a simple just running a PCR assay. Without a proper method or for sample preparation and trying to get rid of inhibitors, no matter how good of your, say, PCR assay is, you are likely to have lower sensitivity and which will impact negatively on the outcome of your result. Okay.
Thank you very much, Dr. Liu. I'd like to ask you 2 more questions before we conclude our interview. First is,
yes.
Do you think that PCR can be effectively engaged in a production setting rather than a laboratory setting?
Definitely. PCR is becoming kind of universally adopted, throughout the world for detection and identification of foodborne pathogens. And I mean, at the moment, it's more like in the clinical laboratory, in the research laboratory. But I feel, in the coming years, the food producers and will probably apply this one and to enhance their the assay for this organism and reduce the probability of the foodborne pathogen not detected and then causing problems to the consumers. Okay.
Very good. We've had a number of challenges in some industries, particularly those that are particularly plagued with foodborne pathogens like sprouts. Can you comment on the potential use of PCR for sprouts?
You mean the spores, or can you just repeat that question?
Because, in the United States, we've had a lot of challenges with either wheatgrass sprouts or bean sprouts and the potential detection of Listeria in those products. Do you see an applicability for PCR in those particular products?
Definitely. Vegetables, the sprouts you're talking about, it's kind of because they're unique structure, like they have this, not very smooth surface. The bacteria may be hiding, between the leaves and all that. So it's kind of problem. But if you are using a very efficient method, you should be able to isolate the organism, or at least get the organism out of the vegetables, and using the molecular method, and which will give you a very sensitive and specific detection. Okay.
Well, thank you very much, Dr. Liu. What I'd like to do is allow you some time to just express your general feeling on the use of PCR for food production in green leafies and other produce.
Okay, and I believe that PCR method will be a very powerful technique for detection and identification of foodborne pathogens, and including the green vegetables and the sprouts and all that, and it will lead to cleaner, safe products for the consumers, and reduce the possibility of this foodborne pathogen getting out of the control and then causing problems in the consumer. Okay.
Thank you very much, Dr. Liu, and wish you a great day down under. I know it's early in the morning for you, and appreciate your participation today.
Thank you very much for the opportunity.
Thank you, sir. Have a great day.
Okay, bye.
Bye now.